Okay, here's what we need to know for the Unit 7 test. We started out with animal development. We looked at the journey from going from a zygote, which is a newly fertilized egg, to the 4-cell stage, to the 8-cell stage, to the blastula, it then infolding and forming that blastopore thing so that it's a gastrula, and then finally then developing into a fetus of some kind. So in the case of a protostome, we know that a protostome, that blastopore forms into the mouth, proto meaning first, stome meaning mouth. And then the deuterostome forms the mouth second, deutero meaning second, so mouth second. But the key here is it's going to form that infolded region, and the infolded region here is going to be either mouth in a protostome or the anus in the deuterostome. Then we went on to talk about the lymphatic system. We said that your lymph nodes are like your security checkpoints in the immune system security system, and so that's where it's going to check for all these antigens. And the reason why that's an important place to check for antigens is because lymph is mostly made out of all the leftover stuff, all the waste from the extracellular space, from that interstitial fluid and extracellular fluid and blood, so it all goes into the lymph, and then the lymph the lymph nodes sort of filter through all that stuff to see if there's anything bad in there, to see if there's any viral particles or anything like that. And when your lymph nodes get swollen, that generally indicates that you have a immune system response going on, and we know that that means that these first parts here, this non-specific immune system, must not have worked out for you. It must not have kept out the pathogen, because your physical barriers, which are skin for the most part, keeping out various pathogens and not allowing them into the body in the first place. You've also got mucous membrane. Mucous membrane is any place that you actually have to have a barrier that you or have to have an opening, rather, that you have a barrier in there that prevents too many pathogens from going in. Then you've got also your stomach, right? The stomach acid acts as a barrier, make sure that everything is dead by the time it gets into your intestines. If something still does end up getting in some ways, let's say if I have a cut and I get an infection, I get a bacteria that gets into that cut, well, that would be handled by phagocytes. And we talked about the different types of phagocytes. You've got neutrophils that are going to handle larger infections, not large-scale infections per se, but large infectious agents, like things like flatworms, for instance. If you had parasitic flatworm, those would be handled by neutrophils. You've got your dendritic cells that are near the surface of the skin. Those are going to cause an immune response, anything that is able to pass through the skin. And then finally, you've got monocytes, which would then differentiate into macrophages. And then once you actually have a site of an infection, the idea is you want to get all of these things to the site of the infection. And so that's going to be where the inflammatory response comes in. Those dendritic cells will release antibodies, specifically these antibodies called immunoglobulins, right? And those immunoglobulins will then cause cells called mast cells in your body to release histamine, and the histamine is going to cause things like vasodilation, meaning your blood vessels are going to get larger, and as your blood vessels get larger, that's going to increase the permeability of those blood vessels, of the capillaries, and so if you have increased permeability, that means that more blood is actually going to get through, more fluid is going to get through, more blood proteins are going to get through those capillaries, and so it's going to lead to things like swelling and redness and increased temperature and also pain caused by the swelling. So those things are good, though, because that means that more of these cellular defenses are actually going to get to the site of the infection, where they can then prevent or stop a larger infection from occurring. We know that allergens are when your body kind of messes up. It doesn't do exactly what it's supposed to do. They're false pathogens. and a bunch of IgEs get released, and mast cells then respond to that, and those mast cells are going to release histamine, and as the mast cells release histamine, that's going to lead to all of these things: swelling, redness, increased temperature and pain that we know from allergic responses. If you have a systematic allergic response, meaning that you have swelling, redness, increased temperature and pain throughout your entire body, the biggest problem is the vasodilation that occurs because your blood vessels get much larger, and as your blood vessels get larger, your blood pressure goes way down, and as your blood pressure goes way down, that can cause you to pass out, and it can cause not enough oxygen to get to the brain, and then you can have other problems that occur, like too much swelling. You can get swelling in your throat and your trachea, and that prevents you from being able to breathe, which is also bad due to decreased oxygen in your body. Once the actual pathogen is identified, though, you have this thing called the specific immune response, and the specific immune response gets divided up into two different sort of subgroupings. There's the humoral immune response. The humoral immune response is when you have pathogens that are not in a cell. They haven't entered a cell yet. They're in your extracellular space. So we call that the fluid that's in the extracellular space, we call the humerus, and that's where it gets its term humoral immune response, because it's in that body fluid that's not in a cell. And these are going to be mediated by B cells. And so we know we start out with a naive B cell, right? That naive B cell is one that is one that has never seen a infection before, and so it's going to be checking for a specific type of antigen, and it's got antibodies on its surface. So those naive B cells, like I said, they look something like this, where they're lined with these antibodies. And these antibodies can interface with various antigens, right? And once they find them, remember you have a very small amount of these naive B cells, but once they find them, they will latch onto them and they will activate, right? They become plasma cells, these activated B cells, and the plasma cells then have to wait for a signal before they can divide. And they can divide up into two different types of cells. Plasma cells divide, and the types of cells that they're going to divide into are called effector B cells and memory B cells. And in order to divide, in order to be allowed to divide, they need these things that are called cytokines. So it requires cytokines. And where do cytokines come from? Cytokines are released by these things that are called helper T cells. So why would a helper T cell then release cytokines? Well, these plasma cells are going to present whatever they've found. So once the plasma cell finds a pathogen or an antigen of some kind, it's going to present that on its MHC2. And as it does that, as it presents that thing on the MHC2, that gets checked by a helper T. And that if that helper T cell then is happy with what's going on, it's happy, it's saying, good job, you found a bad thing, that helper T cell then releases cytokines, which then allows for this to happen, right? So this little thing here, this little aside, is to make sure that there's no plasma cells that are just going to go crazy and do their own thing and start making effector and memory B cells. That's just double checking their work, right? The plasma cell does something and then it checks to say, hey, did I do a good thing or did I not do a good thing? And if it does a good thing, then it gets to divide, and if it wasn't a good thing, it'll go through apoptosis, and we don't have to worry about that plasma cell anymore because now it's dead. Okay. So Then the alternative to this is the cell-mediated immune response. And the cell-mediated immune response is going to revolve around these things, cytotoxic T cells and NK cells. Specifically, cytotoxic T cells have a much bigger role in the cell-mediated immune response, because when we say cell-mediated, what we mean is that a pathogen has entered a cell and that cell is now infected and it's producing some non-self protein. Okay, so you've got these things. So let's say a virus infects a cell. So we'll go down here. Virus enters a cell. Okay, well, what's the first thing that a virus is going to do in a cell? A virus is going to say, hey, cell, I need you to make these proteins for me, right? It's going to inject some RNA or maybe some DNA, and it's going to hijack the ribosomes of that cell in order to produce its own stuff. So the cell begins producing viral particles. Well, the cell doesn't know that it's doing anything wrong, right? The cell doesn't know that it's producing viral particles. It doesn't know where the instructions came from for any particular protein that it's producing, and so it doesn't think anything's out of the ordinary. But as it's producing these viral particles, it's going to tell the rest of the body exactly what it's doing. And so it's going to rely on this thing that's called MHC1, or the major histocompatibility complex 1, where it's going to present whatever proteins are being produced within that cell, right? So cellular protein products are shown on MHC1. Okay, well, these MHC1s are constantly being checked, and they're being checked by these things called cytotoxic T cells. And in the event that a cellular protein product is shown on MHC1 that's not from your body, we call those non-self proteins, a cytotoxic T cell is going to see that, and it's going to say, uh-oh, there's a problem. So the very first thing that the cytotoxic T cell would do in the event of a non-self protein being produced is that this cell goes through apoptosis. So it kills that cell so that it doesn't continue to produce non-self proteins, and if it is able to trigger that response, this is going to then divide up into two different types. It's going to divide up into effector T cells, which again do the same thing as the cytotoxics. They go around and look for the bad stuff. And then also memory T cells. And those memory T cells, those are going to be the ones that are going to look for future infections. So we know memory B cells and memory T cells now, and those are going to be the main component of your lymphatic tissue. So when we said that the lymph nodes are the security checkpoints, well, the memory B cells and the memory T cells are like the machinery or the security guards within those checkpoints that are preventing bad stuff from occurring, right? They're preventing these pathogens from hurting you, from destroying your cells and producing too many of these pathogens. When NK cells come into this mix here is when this step doesn't work out here, when the cell begins producing viral particles, but it doesn't actually show them on MHC1, right? Sometimes what happens is they don't show anything on MHC1, right? And so then NK cells are checking for this thing. So an NK cell can come into the mix here, and if that cell is not producing anything to show on MHC1, then an NK cell will kill that cell, because sometimes these viruses, they'll disable MHC1 prior to producing any proteins, because they know that if the cell tells the body what it's doing, that a cytotoxic T cell will eventually kill it. And so it hides what it's doing by disabling MHC1, and the NK cells are going to be looking for that particular situation. Is MHC1 functioning? If it's not functioning, then that cell gets killed. If it is functioning, then the NK cell won't bother it. The interesting thing about an NK cell is that an NK cell is just checking for the presence of MHC1 or not. It doesn't actually check what's made on MHC1. So if you go and if an NK cell checks MHC1 on an infected cell and there's something on there, then an NK cell actually just leaves it alone. The only time that NK cells kill a cell is if it doesn't show it what it's making, right? But if it's making something bad and it shows it what it's making, it thinks that's fine. So they kind of do opposite type of things. This is looking for bad stuff. This is looking for the lack of stuff. Autoimmune disorders are when your T cells are going to destroy your own cells for whatever reason. Normally if they're just destroying a cellular product like a B cell would do, it's not as big of a deal. So the cytotoxic T cells are the ones that cause major problems when they're actually destroying the cells. Now, the reasons why they do that are varied. It could be that that a B cell triggered this initially, and then cytotoxic T cells, a B cell sees some cellular product and then produces antibodies for it, and that makes it more likely for a cytotoxic T to attack it. That is possible, but generally speaking, if we say that the immune system is attacking one particular cell type, well, guess what? It has to be a cytotoxic T cell that's doing that, because the cytotoxic T is the type that kills cells. That's what cytotoxic actually means. Cyto means cell, so it's a cell that is toxic to other cells, meaning it's going to kill those other cells. We moved on to the nervous system then. Nervous system, we know is mostly made of neurons. There's two different cell types. There's the neurons, which are the important type, the kinds that's actually sending the electrical signals. And then there's all the support tissue in the nervous system that we call the glial cells or neuroglial cells. But really what we focused on were the neurons and the difference between resting and action potential. Resting potential is when the neuron is a salty banana, so it's got sodium on the outside and potassium on the inside. And then once it's triggered, those two ions are sort of going to switch places, and you'll end up with an action potential where that electrical impulse fires down the neuron and then stimulates the next neuron. There are different types of neurons. We know that there are sensory neurons, there are motor neurons, and there are interneurons, right? And so the general path that it would take is if you had a small neuron like this. This would be like a sensory neuron, right? And this sensory neuron would then trigger some other neurons. Let's say it's triggering these neurons here. And these other neurons might have very long axons because these are interneurons. And then at some point, what's going to happen is there's going to be a bundle of neurons that have really short axons like this, and they all do some magic. There's something going on in here. This is like in your brain or something, some gray matter. And then this is going to integrate the signal, and then it's going to send the signal out, right? It's going to send this signal out to something, to an interneuron, and that interneuron then is going to send that signal to a motor neuron, right? So the sense comes in here, it then sends this through interneurons to some space where it's processed, and then that processing signal gets sent out to a motor neuron that then changes your behavior in some way. It flexes a muscle or does something else. Okay. The point here is that this space where you have a lot of axon distance, that's white matter. And then this space where you have not very much axon distance, this is gray matter. The distance in between these neurons, the spaces in between, so like in here, these are called synapses, and synapses are mostly going to be they're all going to be either electrical or chemical. If we're talking about the ones that are in between here, these are, we can assume as part of the peripheral nervous system because of the long axons here. We would say that these are mostly going to be chemical synapses, ones that rely on neurotransmitters, versus these ones in here where you have a whole bunch of them, This would be like in gray matter, and those are mostly going to be chemical synapses. Chemical synapses, electrical synapses rather. Electrical synapses are those ones that use ions, and then chemical synapses are those ones that use neurotransmitters. In both cases, there are inhibitory signals and stimulatory signals, right? Those inhibitory signals would be stopping the next neuron from firing, and the stimulatory system makes it more likely for that next neuron to fire. Inhibitory neurotransmitters, we talked about dopamine. We said that epinephrine is inhibitory towards certain things, like towards pain sensors and also towards your smooth muscle. And then stimulatory neurotransmitters, epinephrine is stimulatory towards sensory nerves, those things that are allowing you to actually observe the world around you. As far as a neuron diagram goes, you can be as fancy or as not fancy as you want. This is how we normally draw them. So you got dendrites up here. Right, these are the parts that are going to receive a signal from the last neuron. This is the cell body here, and what happens is as these neurotransmitters are received onto the dendrites, that's going to cause Ca2 plus ions to enter. As the Ca2 plus ions enter, the interior portion of the neuron, which is normally negatively charged at resting potential, it's going to become more positively charged. And as it becomes more positively charged, that's going to cause all of these gated ion channels to open all along the axon, and it's going to allow sodium ions to flow in. And as those sodium ions flow in, that's going to allow for this signal to be sent all the way down the axon, and then the axon terminal will release neurotransmitters of its own along the next synapse. This is an unmyelinated neuron that we're showing here. The myelin sheath is secreted by glial cells that are called Schwann cells, and what they do is they block a bunch of those gates in order to cause the voltage differential or the voltage jump to be greater from the inside to the outside, and that causes the signal to actually speed up as it travels through these areas where it can get in. These areas where it can get in are called nodes of Renvier, and that's going to allow it to move faster down very long axons like you would find in white matter, where you have a long distance to travel in the nervous system. We talked about some parts of the brain. The important parts of the brain that you need to be able to divide up is to say, all right, in a human, this is a little bit different because we know that our midbrain is technically our brain stem, and our hindbrain is our cerebellum, but that's due to some developmental weirdness that we have, because in most organisms you can divide up the forebrain, the midbrain, and the hindbrain into the most primal and most important of all of the functions. Those are all going to be handled in the hindbrain, and then midbrain is going to handle all your sort of mid-level stuff, like the regulation of food intake and things like that, movement towards or away from a light source. That would be handled midbrain, and then forebrain is going to be all of the voluntary stuff, like can I think about doing something and do it? Can I will myself to do something? And so if I think about like, okay, can I will myself to stop my heart? Well, I can't think about that and stop it, so that must be handled somewhere else, like in the midbrain or the hindbrain. Versus can I move my arm? Yes, I can move my arm. Therefore, that is handled in the forebrain because it's a voluntary action. As organisms get simpler and simpler, where they have less and less control, their hindbrains are much larger than the rest of the brains. We know that humans, having a lot of control over our body and the way that we act and things like that, we have extremely large forebrains and smaller hindbrains and midbrains. As far as your peripheral nervous system goes, anything that's not the brain and the spinal cord is going to be part of the peripheral nervous system. And there are, again, 3 different types of things in the peripheral nervous system. There are motor neurons, there's sensory neurons, and there's also those interneurons in between them. But we mostly think of the sensory neurons and the motor neurons as the thing that actually do stuff, right? Interneurons are like the wiring between those parts. But the sensory neurons are going to give us an idea of the world around us. The motor neurons are going to allow us to respond to the world around us, and then most of these signals have to get sent to the central nervous system to actually do something. These reflex arcs here, they again are always going to move towards the central nervous system, but whether or not that the gray matter, this part that is actually processing the signal, whether that part is in the brain or the spinal cord differs based on the reflex arc. So like the knee-jerk reaction is in the spine, but like your mammalian dive response is in your brain. Both of them are reflexes because they're unlearned, they're innate behaviors, but they are handled in different places, and as a result you have more control over the one that's in your brain because it has to pass through the rest of your brain before it actually gets to that reflex, and then it has to send it back, and it allows your brain to filter out those things. All right, endocrine system. Lots of stuff for the endocrine system. Some stuff you need to know about, some stuff is less important. So you should definitely know the difference between a steroid hormone and a peptide hormone. Steroid hormones are the ones that are fat soluble, and so the receptors are going to be on the inside of the cell because of the fact that the receptors, or because of the fact that the hormones themselves are going to be able to dissolve through the plasma membrane to actually get into the cell. A peptide hormone can't do that because a peptide hormone, being water soluble, is stuck outside the cell, and therefore when we look at the signal transduction pathway, these peptide hormones have to have their receptors on the outside where they can then be able to get to. It wouldn't do any good to have a peptide hormone receptor on the inside of the cell because the hormone would never get to it, and therefore that receptor would never see that hormone. We know there's internal and external hormones. Most of the human hormones are internal hormones because we have communication in other ways. But if we look at other organisms like ants, they have a significant amount of external hormones because that's their main form of communication from one to the other. If it's part of the internal system, we call those endocrine glands. If it's part of the external hormones, we call those exocrine hormones or exocrine glands. The stuff that you should know about, your thymus is important for your immune response. We know that the T cells are going to mature in the thymus, and that's how you can remember the B cells are going to be maturing in the bone marrow, and the T cells are maturing in the thymus gland. The thyroid and the parathyroid are going to control metabolism. So your thyroid controls your metabolism, controls how high the rate of cellular respiration is in your body, and then the parathyroid controls solute concentration in your blood, specifically the amount of calcium in your blood. So a parathyroid disorder would cause there to be too much calcium in your blood, and a thyroid disorder could cause your metabolism to be too high. It would be called hyperthyroidism, or too low would be called hypothyroidism. Pineal gland is going to regulate your circadian rhythm or regulate your sleep and wake cycles. Pituitary gland is going to be controlling other glands in the body, and it's going to maintain homeostasis. It's also going to control your growth. So we talked about pituitary disorders where you might grow extremely tall or extremely large, and those are all generally caused by overactive pituitary glands. Pancreas, we know, is, I don't know why glucose is spelled like that, like with a glue, but glucose regulation is, your blood glucose regulation is your main job of your pancreas. So it's going to release insulin when your blood glucose is too high, and that's going to cause your cells to start taking in more glucose from your blood, right? So they take that blood glucose level and they bring it into the cell, and they use the glucose instead of using it just for cellular respiration, which they always do. They're going to, in addition to that, they're also going to use that glucose to make glycogen. Glycogen is a storage polysaccharide that they can then store for later. It's mostly muscle cells and liver cells that are going to do this. And then later on, if you need glucose, like if you haven't eaten for a while and you're starving, your body then releases the other hormone from your pancreas, which is called glucagon, and glucagon then leads to your muscle cells and your liver cells breaking down that stored glucose that they have, the stored glycogen, and they break it down into glucose and then release the glucose into the blood to raise the blood sugar to allow other cells to use all that stuff. Your adrenal glands are going to be your response to stress. Your adrenal cortex is going to be a general stress response, right? Like some heightened nerves and stuff like that. But then your adrenal medulla, which is the interior portion of it, is going to be things like epinephrine and norepinephrine that are causing that immediate, okay, this is a very stressful situation. It's an adrenaline situation. It's a fight or flight response, and I need to do something. Versus your adrenal cortex that, like I said, it secretes things like sex hormones, but it also is going to release your what are called corticosteroids, and corticosteroids are those ones that are controlling just your overall stress from one day to the next. Okay, we're sick, and therefore our body processes are going to go a little bit faster than normal. They're going to go, our metabolism is going to be a little higher because we're repairing the body, not the fight or flight response stuff. So the cortex is going to be general, and the medulla is going to be fight or flight. Gonads, sexual development, testes in males, ovaries in females. They release the things that cause the secondary sex characteristics in both sexes. Those are testosterones in men and estrogens in women for the most part. There will not be anything specifically on animal behavior on this test, but there may be something on chi-square, because we have done chi-square already. So you should be familiar with the chi-square equation and how to work the chi-square equation. The chi-square formula will be given in the event that you do need to use it. The chi-square formula and what all the stuff means, and the chi-square table with degrees of freedom and confidence intervals or p-values, those will all be given as well. You should know generally what to do with that. If you don't know what to do with that, check out the video from the 22nd of February, which is when we went over what to do with chi-square values. And that's it. You guys have the rest of the period to study.